day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Ditero Magwala, and this program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So in today's lesson, we are still looking at DNA, the code of life. We're looking at our revision. And we are focusing on protein synthesis. So here we will be looking at both phases of protein synthesis, which is transcription and translation. So we're just going to look at that. We're going to look at the process, and we're going to go through some activities. Right. So here, what are we going to be looking at? We're going to be looking at how DNA and RNA are involved in this process. So basically, what is the role of both DNA and RNA in the process of protein synthesis, right? And then we're going to look at transcription, which is the first phase whereby DNA is used to form mRNA, right? So just there we are already finding the role of DNA in the whole process, right? So it plays a role in transcription whereby it is used to form the mRNA molecule, right? So here we are going to look at what exactly happens. We're going to look at the double helix that unwinds. We're going to look at how the DNA double strand unzips and breaks, right, as the weak hydrogen bonds break, right, and then to form two separate strands. Now, remember, one of these strands, not both of these strands, but only one of these strands is going to serve as a template. So one strand serves as a template for the formation of mRNA, and this uses the free RNA nucleotides. Now, remember, these nucleotides are the building blocks of both DNA and RNA. So the RNA nucleotides would be the building blocks of RNA or mRNA. And then this mRNA is complementary to the DNA strand that is serving as a template, right? And the mRNA now has the coded message for protein synthesis. This message is the message that was coded for by DNA. Right, so the DNA molecule plays a role by coding for the message that is needed for protein synthesis. Right, so basically DNA controls protein synthesis because whatever DNA strand you have is going to determine the kind of protein that you have, right? Then next, we know that the DNA molecule, the mRNA molecule is going to move out of the nucleus, right? It moves out of the nucleus through the nucleopores into the cytoplasm. And it reaches the cytoplasm, it attaches itself onto the ribosome, whereby translation is going to take place. Now, what happens during translation? Each tRNA will carry a specific amino acid, and this tRNA will be carrying the amino acid based on the anticodon. So when the anticodon on the tRNA matches the codon on the RNA, we're going to look at that on the mRNA, right? Then this tRNA brings the required amino acids to the ribosome, right? Okay, the amino acids, remember, these are the building blocks of the protein, right? You don't have to know the names of the codons or the amino acids that they code for. Remember, you'll be given that table whereby you are going to compare the amino acids and the codons or anticodons that you need to work with, right? And then the amino acids will become attached to each other by peptide bonds. The bonds that bring the amino acids together are the peptide bonds, and that will form that required protein, right? Okay, so that is what we're going to look at. However, as you are looking at this is just how you should be able to describe both translation and transcription. So if you just write this down and you learn this, then you know how to describe the whole process of protein synthesis. So this is the description of transcription, and here we have the description of translation. Okay, we are still going to look at this in a lot more detail so that we can understand everything. Right, now remember our paper, the structure of our paper. This is very important, guys. Remember that this is a 2.5 hour paper and it is for 150 marks. Now, just a reminder of how the marks are broken down. Section A and Section B, only two sections. In the past, we had three sections, section A, B, and C. Do not prepare for section C anymore. Just a reminder of that. Section A is 50 marks. Here we have our short questions, multiple choice, terminologies or biological terms, and the matching column whereby you match column A with column B, right? And then section B, we have question 2 and question 3. Each would be having 50 marks, and those would be the long questions. Okay, now what should you be able to do by the end of today's lesson? You should be able to describe transcription. You should also be able to describe the movement of mRNA out of the nucleus. And lastly, you should be able to describe translation, right? You should also be able to use the given codons and anticodons to match with the given amino acids in order to look at the coded message for the formation of a protein. Okay, now let's have a look at our examination tips, the things that you should just take the knowledge with when you go into the examination room in order for you to be able to answer those questions with as much knowledge as possible, so that when you answer with the purpose of making sure that you are doing exactly what the examiner or the marker wants, right? So let's have a look at these marking principles. So these marking principles are the principles that are used by markers, right? As your paper is being marked, these are some of the guidelines that are being used in order to make sure that everything is marked accordingly. The first one says, if more information than marks allocated is given, stop marking when maximum marks are reached and place a wavy line. So you place a wavy line and write max for maximum there, right? So this is just for you to understand that no matter how much even if you give a lot of information, if that information is not needed, you will not get extra marks. So if a question is 5 marks and you give a whole page of answers, once the 5 marks has been reached, the marker will stop reading and indicate that the maximum marks have been reached, right? If, for example, 3 reasons are required and 5 are given, mark only the first 3, irrespective of whether all or some are correct or incorrect. So the marker will not go around your answers looking for the correct answers and ignoring the incorrect answers. The first 3 will be marked. Even if number 1 is incorrect, number 2 is correct, and number 3 is incorrect, and the remaining 4, 5, 6 are correct, you are not going to be marked based on the correct ones. You're going to be marked based on which ones came first. So make sure that you do not just throw in answers there with the hope that the marker will look for marks and give the answers that you are sure of. And this will also save you time, because you do not need to be wasting your time giving more answers than needed. If the whole process is given when only part of it is required, read all and credit the relevant part. So here you will not lose marks for giving a bit of extra information. However, refrain from that if you can, so that you do not waste your time. Here, 150 marks, 150 minutes. It means 1 minute, 1 mark. And then number 4, if comparisons are asked for and descriptions are given, accept if differences or similarities are clear. I think this one is straightforward. If you are asked to give similarities or differences and you do not give straight answers, you will only be marked if within your description the differences and similarities are shown. But remember, if this is a tabulate question, you will lose a mark for not tabulating, which is our 5th point. It says if tabulation is required but paragraphs are given, candidates will lose marks for not tabulating. You cannot lose this 1 mark for tabulation. That is a free mark. So make sure you always tabulate. If diagrams are given with annotations when descriptions are required, candidates will lose marks. This one is a no-no. Make sure you never do this, because you will lose a lot of marks. Descriptions are usually about 5 to 6 marks. Now, if you draw a diagram for a description, you are going to get 0. So make sure you do not do this at all. If flowcharts are given instead of descriptions, candidates will lose marks. Number 6 and number 7, never ever do this, because you will definitely lose those marks. Caption: all illustrations, graphs, diagrams, and tables must have a caption. Make sure you have a caption. Make sure you have a correct caption. Especially when you're drawing graphs, make sure that your caption includes both variables, both your dependent and your independent variable, and it must be correct. If there is a caption but it is incorrect, it will not be marked right. The caption does not get marked simply because it's there. And then if only the letter is asked for but only the name is given, and vice versa, candidates will lose marks. Make sure that you give what you are asked for. And also, if the question says give the letter and name, give both the letter and name, because that is 2 marks. If you give one, you will only get that 1 mark. Okay, now let's look at the specific tips for our protein synthesis topic for today. So you should be able to distinguish between the triplet, a codon, and an anticodon. Remember, these 3, this is the triplet on a DNA molecule, and then codon, mRNA, and then anticodon would be the tRNA. So you need to distinguish between these so that when the question says tRNA or codon, you know what they mean. When the question says anticodon, you know what they are referring to. So make sure that you do not mix these up, right? It helps a lot to know this so that when the question is talking about translation and transcription between these 3, you understand what you should do. And then you should also know where and why transcription and translation takes place. Transcription takes place in the nucleus in order to form mRNA. Translation takes place in the cytoplasm in order for the formation of the protein, right? Know that mRNA is formed using DNA in the nucleus. And then you should also understand that during transcription, adenine will pair with uracil. So you should not pair thymine with uracil, because you usually see when you go through some study material, it would say that uracil replaces thymine. It means that uracil will pair with adenine instead of thymine pairing with adenine. So whatever pairs with adenine would have been replaced here. So that means that you should not say, okay, wherever I see uracil, I will pair it with thymine. Make sure that you do not pair thymine with uracil, but wherever you see adenine, you pair it with uracil instead of thymine, especially when you are having a transcription from DNA to RNA. During translation, adenine pairs with uracil again. mRNA and tRNA do not contain thymine. That is just a reminder there. Right. Okay. And then when we look at mutations in protein synthesis, remember mutations can happen whereby the DNA sequence changes, right? Whereby you end up with a different codon than what was supposed to be there. Once you have a different codon, we know that it means you have a different anticodon, you'll have a different amino acid. Therefore, it could mean that the protein formed is different. Now, this is not always the case. Mutations do not always lead to a different protein. Now, how is that possible? See, if the sequence of the mutated DNA still codes for the same amino acid, the protein formed will remain the same. So remember, if you have a codon, an anticodon, or a triplet, let's say you have UC, and it gives you cysteine, for example. And then this UUC is mutated and you have UCC, right? So the U has changed to C, but it could still be coding for cysteine. Now, if that is the case, it means that nothing much has changed. The same amino acid still applies. The amino acid sequence is still the same. Therefore, the protein formed will still be the same. So you should not cram that every time you see a mutation, then the protein is different. Just go through it and understand exactly what is happening. But what you would see when this is happening, you would see that on the table of amino acids that you're provided with, the amino acid would appear twice with 2 different codons or anticodons. So make sure you check so that you are sure of that, right? So the protein will change if the new sequence of the mutated DNA codes for a different amino acids. But if it does not code for a different amino acid, then the protein will not be different. This means that more than one codon can code for the same amino acid. All right, now let's have a look at our terminologies. Amino acids, those are the building blocks or the monomers of a protein. Protein synthesis is the process during which proteins are made. And then we have transcription, which is the process that occurs in the nucleus when mRNA is formed from a DNA strand. And then we have translation, which is the process that occurs in the cytoplasm during which amino acids combine according to the sequence or the order of the codons on mRNA. And then what is the codon? That is the triplet nitrogenous base that is found on mRNA. Anticodon, the triplet nitrogenous base found on tRNA. Right. Okay, so we have come to the end of our tips for protein synthesis. Now let's look at proteins. What are these proteins? How are these proteins formed? Right. So we know that these are organic compounds, right? And they are made up of amino acids, which makes the amino acids the monomers or the building blocks of proteins, just as we've seen in our terminologies, right? So we know that these amino acids are going to be attaching with each other. One amino acid after the other, they are going to attach to form this long chain of amino acids that would then be our protein, right? So they attach to each other by a process of dehydration synthesis whereby water is lost, right? And the bond that holds these two amino acids together is called a peptide bond. So amino acids are held together by peptide bonds. Okay, so when we have two amino acids, they form a dipeptide, and then if we have 3 or more amino acids, they form a polypeptide chain. Now that polypeptide chain will become so long that we will then refer to it as a protein. Okay, how many amino acids do we have? We have 20 different amino acids, right? So just like we have 26 alphabets from A to Z, we have 20 different amino acids. But remember, just like with the letters, we can have maybe the word together. As we can see here, the letter E appears twice. However, we still have 26 letters, different letters, but the letter E appears twice here. So that is how amino acids are also going to work. Of these 20 amino acids, some of them are going to be repeated or to be used many, many times as this long chain is being formed to have a protein. However, the amino acids are only 20. So we can also see the letter T is appearing twice again. So that means that to form the letter together, the word together, T was used twice, E was used twice. However, it's still E and it is still one T, right? Okay, so all these proteins are made up of a combination of those 20 amino acids, right? So there's just going to be that repetition of amino acids until we have that specific protein that has to be formed. Now, just a reminder of the sequence in which the process of the formation of the protein is going to take place. You start off with your DNA molecule. This DNA molecule will undergo transcription in order to form your mRNA. And then this mRNA will then be involved in translation, whereby we're going to look at the formation of the amino acid sequence or that polypeptide chain. So this is what we would refer to as a polypeptide chain, right? These amino acids. Now, as I was talking about the letters, the letter E and the letter T, which we're repeating, we can see here this color is repeating, meaning that this is the same amino acid. However, it has repeated. This one also, it has repeated. So this is the same amino acid, right? So of those 20 amino acids, we can already see that 2 types of amino acids are repeating themselves, right? So that they will contribute to the formation of that long chain of amino acids, which would then form a protein. So we can see the amino acids there, they form that peptide. Again, the amino acids are repeating, and then eventually we will have that protein that is made up of a large number of amino acids. Okay, and then this is just that example of the 26 letters in the alphabet that will make up different words. So the 20 amino acids will make up different amino acids. I mean, the 20 amino acids will make up different proteins. Okay, and then just as we were looking at the complementary base pairing in DNA replication, complementary base pairing still applies here when we look at the formation of mRNA from DNA. During transcription, we're still going to apply complementary base pairing, whereby we can see that this chain here, which is a DNA molecule, it will be the one that acts as a template. Remember, only one will act as a template. And as you can see, this thymine is pairing with adenine. Adenine is pairing with uracil. It's pairing with uracil and not thymine, right? Adenine is pairing with uracil. It's not pairing with thymine. Why? mRNA or tRNA do not have thymine. And then we can see the C with the G until we have this mRNA chain, right? And then this will then be used for the formation of the polypeptide chain, whereby the amino acids will be joining each other based on the anticodons on the tRNA. We will look at that in the next few slides. Okay, so here we have three phases of protein synthesis: transcription, which takes place in the nucleus. We have the movement of the mRNA out of the nucleus. Remember, mRNA was formed here. mRNA was formed during transcription. It moves out of the nucleus into the cytoplasm, and then when it reaches the cytoplasm on the ribosome, it is going to undergo translation. So those are the three stages of protein synthesis. Let's have a look at the first one, the first stage, which is transcription. Right, so there we can see our nucleus, we can see our DNA molecule inside the nucleus, and it will undergo certain steps in order to form that mRNA. Right, we can see here the mRNA has been formed, right? So what happens? It starts off as a double helix. That double helix will unwind. As the double helix unwinds, the weak hydrogen bonds will break between those two strands of DNA. Remember, DNA is double-stranded, isn't it? So the two DNA strands will then unzip and they will separate. As they separate, one of those two strands that have separated will act as a template. The other one is not useful. So this one is not being used. However, this one is our template. Right. So one strand of DNA will act as a template for the formation of the mRNA. How does this mRNA become formed? It becomes formed using the free RNA nucleotides from the nucleoplasm, which will then attach to their complementary DNA bases. Now let's have a look at that. So for example, here we can see that the green is representing adenine, and then the red is representing thymine. So thymine pairs with adenine, and then we can see here, what do we have here? It's adenine in green. It is pairing with the orange, which is uracil. So you will see that color shift there. If you look at the green and the yellow and the blue, it's always yellow and blue because it's always cytosine and guanine. However, here, adenine, thymine, and uracil, somewhere you'll see uracil, somewhere you'll see thymine. When you see thymine, again, that is the guide for you to say this is a DNA molecule. When you see uracil, it is a guide for you that this can either be a tRNA or an mRNA. All right, and then again, remember, three adjacent nitrogenous bases are called a triplet. That is our DNA triplet in DNA, right? So there we have our triplets. We have 1, 2, 3 triplets. These 3 triplets are going to act as templates for the formation of the mRNA codons. Hence, we have these 3 mRNA codons. We have this, whereby the pink pairs with the yellow, the blue pairs with the orange, pink with yellow, as you can see. And then remember, you will see that somewhere, somehow, adenine will pair with uracil, or uracil will pair with adenine without any thymine. Right, okay. And then 3 adjacent nitrogenous bases form a codon. We've already spoken about that. And that process of transcription is always controlled by enzymes. Let's have a look at our first activity. I'm going to give you that 5 minutes for you to attempt these questions on your own. Remember, here you are preparing yourself for the exam. So you're going to attempt this honestly. So you're going to attempt this without trying to copy from your notes or anything. Just go through it so that when we go through it together, you can just fix those mistakes here and there. So it says, name and describe the process in protein synthesis that is responsible for the formation of mRNA. Okay, take that 5 minutes and go through this on your own. Okay, we're back. Let's have a look at this activity together. So the question says name and describe. It means that you must name the process and also describe the process. It means you're going to get a compulsory mark for naming. If you only describe, it means you will automatically be marked out of 4, because the 1 mark is strictly for naming. This also applies if the question says, describe the process that is responsible for the formation of mRNA. It didn't say name. However, if you name, you will still get that mark. Okay, let's have a look at that. Naming, 1 mark. Transcription. Transcription is the name of the process. Now, what happens during this transcription? The double helix unwinds and unzips as the weak hydrogen bonds break to form 2 separate strands of DNA, right? 1 strand acts as a template to form mRNA or for the formation of mRNA. How does this mRNA get formed? It gets formed using the free RNA nucleotides from the nucleoplasm. And then this mRNA is complementary to the DNA molecule, right? The coded message for protein synthesis is copied onto mRNA. Now, when you're looking at this question, you must make sure you see exactly where the ticks are placed. Where the ticks are placed, that is where the marks are. As you can see, unzipping gets a mark, unwinds get a mark, 2 separate strands gets a mark, acting as a template also gets a mark. So don't just go around giving answers without understanding exactly what you are giving. So make sure you give the answer, you give the facts, you give the important points so that you score your marks. Okay, second activity. The diagram below shows the process of protein synthesis. So we can see here we have process A, we have process B, we have W, X, Y, and Z. Remember, go through this and just label before you can even start answering your questions. So process A, it will be transcription. B, it will be translation. W, it is a DNA molecule, as we can see. X, that is the mRNA that was formed in the nucleus. Y, this is an amino acid that is carried by the tRNA. So this is our tRNA. Y is the amino acid. And Z is the ribosome. Okay, now let's have a look at the questions. I am going to give you a few minutes also to go through the questions. I'm going to give you about 6 minutes, 5-6 minutes to go through these questions on your own, and then we will come back and discuss the questions together. Okay, let's have a look at the questions together. As we are going through these, you are going to be comparing the answers that we are going to discuss now. You compare them to your answer, then you are going to make the necessary changes or the necessary corrections. Okay, 2.3.1, it says, identify the following molecules, W and Y. So we go back to what we've already labeled and we check W. It is the DNA molecule. Y, it is an amino acid. So you are quickly just going to give the answers because you've already worked that out, right? DNA molecule and amino acid. We have the answers for those. The next one says, name the part of protein synthesis indicated by process A. Process A, if we have a look at our diagram, process A is transcription. Remember, the first phase in the nucleus is transcription. The second phase in the cytoplasm is translation. Transcription and translation. Okay. Now, if you know that there's transcription, but you don't know where it takes place, it means that you will not be able to identify it. So you must know where it takes place first. It takes place in the nucleus. So that is why process A is transcription, translation in the cytoplasm. That is why it is said to be taking place in the cytoplasm. 2.3.3, it says, describe how a mutation on molecule W will affect the structure of the protein formed by process B. So how will the mutation that could take place on the DNA molecule lead to or affect the structure of the protein that is formed by process B? If there's any change in the DNA molecule, it means that the protein formed is going to be different. Because remember, the DNA molecule is the one that controls the process of protein synthesis, is the one that controls the protein that is formed. So whatever change happens to the DNA molecule, there's going to be a change on the mRNA molecule. If the mRNA molecule changes, the tRNA changes. If the tRNA changes, the amino acids brought in by the tRNA will be different. And that means that the protein that is going to be formed at the end of the day is also going to be different. But remember what I said when we looked at the tips. I said that this is not always the case. If the amino acid that is coded for can be coded for by two different triplets or two different codons, and that is what has happened, then the amino acid are going to remain the same. The sequence will remain the same, meaning the protein formed is going to be the same. However, in this case, we are going to speak about it as if the change has happened and the amino acid is different. However, if the question you are given is specific and you are able to see that there are no changes, do not just say that amino acid formed, protein formed will be different. Apply your knowledge to that. So in this case, we are giving this answer in a general manner, whereby we will say that the sequence of the nitrogenous bases on the DNA molecule or on molecule W will change. So remember, we were expected to label molecule W, right? And if you did not get that mark for labeling molecule W, you can still say molecule W and you will still get a mark because there was a mark previously for saying molecule W is DNA. You cannot lose marks twice for the same thing. So even if you are not sure, simply say molecule W. You'll get a mark for that. This would cause the corresponding change on the molecule X. Molecule X is mRNA. Also, you can still get a mark for that. And the amino acid brought in by tRNA will be different. And that means that a protein that is formed will also be different. Okay, now we have our mRNA. We will look at the fact that the mRNA will move out of the nucleus. How does it move out of the nucleus? It moves out of the nucleus through the nuclear pores of the nucleus into the cytoplasm. Remember, in the cytoplasm, it will attach itself onto the ribosome, or it will lie on the ribosome. What we are talking about here is what we can see on this diagram, whereby we can see that the double-stranded DNA is forming the mRNA. This mRNA then leaves the nucleus into the cytoplasm. Then in the cytoplasm, now translation starts, whereby we use this mRNA to code for the tRNA anticodons, which will bring the relevant amino acids onto the ribosome to form that long polypeptide chain that will be formed based on the codons. And then as the chain is being formed, we can then say that we have an amino acid sequence and a protein is formed. Okay, now here we have translation. Let's just take a moment here and look at all these labels that we have. These are those labels that will score you very quick marks. It will score you marks. Most of the papers that you are going to be given, you will have this. It might not be translation, it might not be transcription, but labeling, just giving labels, just saying this is a protein, this is an amino acid, this is a ribosome. You will get marks for this, especially in section A, because we don't only have multiple choice and terminologies in the matching column. We also have short questions whereby you are answering questions like this. So just have a look here. This is the mRNA, here is the mRNA. Right? And then here we have the codon on the mRNA, codon on the mRNA, anticodon on the tRNA. And then we have 2 amino acids. These 2 amino acids are joined together by a peptide bond. Right? And then as they are joining by this peptide bond, they are going to form a protein. And down here we have that ribosome, and this would be the process of translation. Okay, so here the tRNA is involved in translation, and at the base of the tRNA, which is down here, that is where we have the 3 adjacent nitrogenous bases, which are called the anticodons. Anticodons are found at the base of the tRNA molecule. Okay, now let's have a look at translation. This is what you would then need to know in order to answer these questions. Now, as you are just going through your preparation, make sure that you go through your past papers, you have a look at your notes, and you write down each and every process that you will come across in life sciences, especially in paper 2. There's a lot of processes in paper 2. We can be looking at transcription, we can be looking at translation, we can be looking at all those processes that you'll be looking at in evolution, your speciation. Just write them down. Make sure you write them down somewhere as you come across them in the marking guidelines, in the memorandums of your past papers. And as you go through that, you will see that it'll be very easy to remember when you get into your examination, because you have seen them, and you have seen them from a memorandum of the past papers. Remember, the process will not change. The process remains the same. So if you just know it and you understand it, then you are done. Also, by simply understanding this process, you're going to be able to answer many other questions that are related to the process itself. So let's have a look at this. Firstly, the tRNA picks up the free amino acids from the cytoplasm, right? So the anticodon on the tRNA will match with the amino acid. Remember, the amino acid that is being picked up, it's being picked up based on the tRNA codon, because this tRNA codon codes for a certain amino acid, and this tRNA codon is anticodon is also dependent on the mRNA codon. So the tRNA takes the amino acid to the ribosome. At the ribosome, the tRNA attaches to the mRNA, a specific mRNA, using the nitrogenous base template rule, whereby the A will be paired with the U and the C will be paired with the G, right? So the codon on mRNA will pair with the anticodon on tRNA. That specific tRNA will be carrying a specific amino acid, which takes us back that this amino acid depends on the type of mRNA and essentially depends on the type of DNA. Right, because the DNA determines the mRNA determines the tRNA, and the tRNA determines the amino acid. So that is why we can say that DNA controls protein synthesis. Right, next, here you will see that we are going to look at the fact that the amino acids are going to attach with peptide bonds to form that protein. So we can see here on the ribosome, we have our mRNA, we have the codons on mRNA. 3 nitrogenous bases, they form a codon. 3 form a codon, another codon, until we get to the end here. Right. Now, all these codons are going to be pairing with specific anticodons. As you can see here, AAA is going to pair with UUU. Now, what amino acid does UUU bring? It brings amino acid lysine. It means that even if we come across another AAA, then we will have another lysine amino acid coming in. Right. So here we have methionine, which is brought in by the tRNA that is carrying the anticodon UAC. Next one, phenylalanine. They will attach to each other by those peptide bonds until we have that long chain of a polypeptide chain, which will then be our protein. Okay, so here we have a diagram that is just representing the whole process, right? Starting in the nucleus, we have transcription. Okay, this transcription, one strand will serve as a template. In this case, our template is the bottom strand. We can see the free RNA nucleotides coming in and pairing. Remember, they don't pair randomly. A pairs with U, C pairs with G. You will see that. And then once the mRNA has been formed, it leaves the nucleus into the cytoplasm. What happens in the cytoplasm? Translation. When this translation happens, here we have our mRNA codons, which are going to be pairing with the anticodons on the tRNA. These anticodons or this tRNA also brings in the amino acids. Certain codon or certain anticodon brings in a certain amino acid, and then they are going to form that chain, as we can see, and then we will have our protein. So you should also be able to label that this is a tRNA, this is an amino acid, these are amino acids on top there, and then this is your mRNA, this is your ribosome as it has been labeled there, and then here we have your DNA molecule, and we have the mRNA leaving the nucleus into the cytoplasm. Let's have a look at activity 3. It says the diagram represents a process during protein synthesis, right? Again, label these, give labels for these, right? Okay, here we have our ribosome at A, we have our mRNA at B, at C, we have our tRNA. D, now, don't cram that. Previously, we saw the tRNA bringing an amino acid from the top. Now, we can see that the anticodons will be here, right? This is the anticodon, meaning that now the amino acid can be at the bottom. So this is our amino acid. And there we have the bond between two amino acids, which is a peptide bond. Now you have labeled your parts, you move on to answer the questions. And you must read this information also, right? Okay, let's have a look at what the next part has for us. So we have some questions. We have 6.3.1 and 6.3.2. We also have 6.3.3. Now you are going to have a few minutes to go through the questions, and then once you are done, we are then going to come back and go through the questions together so that you can make some corrections quickly. Okay, we are back. Let's have a look at the solutions for this. There we have our diagram once again. The first one question says, identify the process above. So the process above, because we can see tRNA, we can see the ribosome, we then know that we are looking at translation. So the process that is shown above is the process of translation. Okay. And then 6.3.2, it says, identify organelle A, molecule B, and bond E. Right, let's go back and have a look. Organelle A, it is the ribosome. B, mRNA. Bond E, that peptide bond. Right, so A, what do we have? The ribosome. B, the mRNA, and then C, a peptide bond, or you can say a peptide. Then that is your marks for that question. And then 6.3.3, it says give only the letter of the molecule that carries the amino acid. Okay, so what carries the amino acid? We already know that what carries the amino acid is tRNA, but you must go back and have a look at what tRNA is. Okay, we'll go back and check. And then the next one, it says the molecule that is copied from DNA. What molecule is copied from DNA? It's mRNA. So we're going to check for tRNA and mRNA. The next one is a monomer. Once we are talking about monomers here, we're either talking about an amino acid or we are talking about a nucleotide. In this case, we don't have nucleotides. It means that we are looking at amino acids, and amino acids are indeed the building blocks of proteins. So we are going to look at the letters that are representing tRNA, mRNA, and amino acids. Now let's go back and find those. Make sure that you give what you are asked for. The question said the letter. So you go back and find out what is the letter that is representing these three. Okay, let's go back. Now we have tRNA. tRNA is represented by letter C. Amino acid by letter D, and the other one, that the answer was mRNA, is represented by letter B. So our answers would be C, B, and D. Let's go and have a look at that. So there we have it. The letter C is represents tRNA, B represents mRNA, and D represents the amino acid. Okay, let's have a look at our last activity. We have the process again. As we can see, we have two processes and some molecules. It says the diagram shows two processes that take place in all cells of the body. Okay, and then we have our questions. Let's go through the questions and then we will discuss the answers. Okay, let's quickly just label. Already we know that process A in the nucleus is transcription, process B is translation, molecule C, as we can see, it is carrying those amino acids and those anticodons. That would be our tRNA. Okay, 3.1.1, it says provide a heading for the entire process illustrated above. The entire process. As we can see there, we have both transcription and translation. It means that the heading for this would be protein synthesis, right? There is your mark. So some of these questions, they are really just giveaway questions. You already know that this is protein synthesis. So you should actually be able to answer this. And as you can see here, they didn't mention that it's protein synthesis because you're supposed to get a mark for it in the questions. And then 3.1.2, name processes A and B. We've already seen that process A is transcription and process B is translation. Okay. 3.1.3, briefly describe the role of molecule C in process B. What is the role of molecule C in translation? First, what is molecule C? Molecule C is tRNA. So what role does tRNA play during translation? We know that it brings in the specific amino acid based on the anticodons that are complementary to the codons on the mRNA, right? So how are you going to structure this? How are you going to put this answer? Remember, it's not always about the knowledge that you have. It's also about how you give the answer. So it is very important that as we are going through these, you check how the answer was given, you compare to your answer, and you make those changes. So firstly, molecule C. The question did not say the role of tRNA. It means you will get a mark for saying what molecule C is. Molecule C is tRNA, and tRNA carries a specific or the required amino acids to the ribosome. When the anticodon is on the tRNA, matches to the codon on the mRNA. And that's it. tRNA brings in amino acids based on the anticodon on the tRNA that is complementary or that matches with the codons on mRNA. That's it. The main words: codon, mRNA, anticodon, tRNA, required amino acid, and the name of the molecule, and also the fact that it brings it to the ribosome. So you can get a lot of marks here. You can actually even get a mark for anticodon on tRNA and then codon on mRNA. That will be 1, 2, 3, 4, 5 marks, and this was out of 3. So you can easily get a lot of marks here. The last part of our question and our activities for today's lesson, 3.1.4. This is the part that I wanted us to discuss, that you should be very careful of. Right, and this question comes up a lot. It comes up a lot. It says here, the table below shows different tRNA anticodons. It's already showing you, it's already guiding you. tRNA anticodons. Remember, this table can either say tRNA anticodon, mRNA codon, DNA triplet. You must be careful. You must check what is it giving me. Then based on this, you will know how to work around what you are given. Okay. The first question says, provide a label for the chemical bond D. It's taking us back to that diagram. Remember, it's still part of the previous question. So chemical bond D here on the diagram is the peptide bond. There is the peptide bond between E and F. Right? So that is the answer for that part. That is a peptide bond. Okay, let's look at B. It says, identify amino acids E and F respectively. So we are going to identify amino acid E and amino acid F. Based on this, we're going to go back and look at the anticodons on E and F. Then based on the anticodons on E and F, we will identify those amino acids. Now, we will look at what we are given there. Are we given the codons or are we given the anticodons? If we are given the codons, we have to now translate them to anticodons so that we can match, because we are given an anticodon and amino acid table. But if we are given anticodons on E and F, that side, then we are just going to take it as it is and compare to the amino acids that we have. Let's have a look at that quickly. Okay, for E, we're given both, right? So the anticodon here is GGC. That is for E. Then we will look on the table what GGC gives us, and then UGC for F. UGC. So we're going to look at GGC and UGC. Let's have a look at that. Okay, GGC, there we have it, and then UGC, there we have it. So our amino acids would be proline and threonine, right? So here, proline, and then F, threonine. It said we must give the amino acids there, right? And then let's have a look at that. Okay, there it is, right? Proline and threonine. And then the next question, 3.1.5, it says, provide the DNA triplet code that would have coded for codon G. Let's have a look at codon G. Here is codon G. It's a codon, therefore it's on mRNA, right? Codon G is GCC. This is mRNA. The question wants the DNA triplet that helped to form this. So what was in here that helped to form this GCC? GCC, we know that G pairs with C, G, G. So this is our DNA triplet. So we have CGG as our DNA triplet. So that is what we would get for 2 marks. Okay, let's have a look at the last question, 3.1.6. It says, if the DNA triplet code that resulted in codon G were to undergo a type of mutation that was mentioned in 3.1.6a, and the DNA triplet changes to CGT, then explain the effect that this would have on the resulting protein. Right, now, if codon G, let's have a look at codon G once again. Let's have a look at codon G. Codon G is GCC. This is the codon. What is the anticodon? Which is what we have on the table. Remember, we don't work with codons on this table. We work with anticodons. So you must first find the anticodon. We have anticodon CGG. What is anticodon CGG? It gives us amino acid alanine, right? So it gives us alanine. That is codon G. Right. The question now says, if there was a mutation, and this mutation led us to CGT. Now we have CGT instead of CGG. Right? Instead of CGG, we have CGT. This CGT, what will it now give us? It will now give us something different. The CGT will now, let's have a look again. CGT will now give us GCU, right? Remember, this is DNA. This is DNA. This is mRNA. But because we are given an anticodon table, it means we must transcribe it to tRNA, which will give us CG. No, this is A. Sorry, this is A. Remember, thymine pairs with adenine. Don't forget that. Thymine pairs with adenine. Therefore, this adenine will pair with uracil. So our tRNA is now CGU. It means it's going to give us a different amino acid. But let's check. Is it really giving us a different amino acid? It's still giving us CGU. So CGT, GCA, CGU gives us alanine. So the amino acid has not changed. The anticodon has changed, but it still codes for the same amino acid, which is what we spoke about earlier when I said the change in the sequence does not always lead to a change in the amino acid. Therefore, we have this, which means that the protein formed will not be affected. We are going to have the same protein. However, there is that change in the amino acid, the sequence of the nitrogenous bases, but the amino acid has not changed. So you must keep that in mind. You must always make sure you check first. Don't just rush in to say the amino acid has changed and so on. Just check first. So codon GCC on the mRNA has changed to GCA. Anticodon CGG has changed to CGU. Right? You get marks for that. Both these anticodons code for alanine. So they code for the same amino acid. Therefore, the protein will not change, meaning that there will be no effect. The question said, explain the effect. If they were giving us a different amino acid, you would say they have led to a different protein formed. But in this case, the protein will remain the same. Therefore, there is no effect. Okay, have a look at the homework, go through the homework, and then you will discuss the answers in the next session. Okay, this is a question whereby you are going to be describing translation. So have a look at that in order to prepare for your examination on that. Now, in summary, we are just looking at the whole process here, whereby we look at the unwinding of the DNA molecule to form the mRNA. We are looking at DNA being used to make a copy of mRNA. This mRNA, remember, will then leave the nucleus into the cytoplasm, whereby translation will take place using the mRNA. The tRNA will bring in the required amino acid based on the sequence of the mRNA. And then a certain protein will be formed, or a specific protein will be formed. This then tells us that the DNA that we initially started off with is the one that controls the whole process of protein synthesis. It controls the protein that was formed, as it controls the mRNA that was formed, and subsequently controls the tRNA that was formed and the amino acid that the tRNA brings in. Okay, now just a reminder, bring all of these things into the examination room with you, so that you have a smooth examination. You do not panic when you come across a calculation or anything that may require this. You need your mathematical instruments, you need your compass and your protractor in case you have to draw a pie chart. You need your ruler to underline and draw graphs and tables, your calculator for calculations. Make sure you bring enough pens so that in case one of your pens stops writing, you can still carry on with your examination. Eraser and pencil as well. So in the next lesson, we'll be moving on to meiosis. We are going to be looking at the process of meiosis. We are going to be just looking at each process and going through the processes that happen in each of these phases. Okay, we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.